your own antigens. So it does not really activate. Okay. However, scientific proof of danger theory were largely lacking. Okay. Subsequently, it was shown that this is uh, actually a very active process, and the response of these potentially autoreactive cells is predominantly dampened by canonical cells that are also passing through this process of positive and negative selection. Okay, basically these are the potential Treg precursors, and they also have to pass through this process and eventually give rise to these Tregs. These Tregs are basically regulatory in nature, and their predominant function is to control autoreactive cells. Right now, later on, actually, many more functions of these Tregs were also discovered. Whether this is not only for T cells, this is also for antibody responses, for innate immune cells, and many more functions. We also know that there is something called as plasticity in Tregs. Treg can also sometimes become potentially reactive, and they start damaging your own tissues, depending on what environment was created following infection, immunization, or because of the homeostatic processes. Okay, all those things are detailed. Okay, it was shown that if you somehow disrupt the function of these regulatory T cells, either by a genetic removal of FOXP3, or if you somehow eliminate these cells once they are formed, there are different ways you can achieve that. Okay, so these autoreactive cells, they start proliferating more, and as a result of it, animals or individuals, humans even, so they start generating what we call as autoimmune reactions or multi-organ inflammatory processes. Okay, these are two examples here. Scurvy, as I said in previous class, was observed in mice. So basically, these animals are called scurfins. Okay, scurvy mice. And IPEX is a hyperinflammatory syndrome that is because of disrupted or a mutation, actually a single mutation in FOXP3 locus, right? Now we are going to talk about actually how these processes actually are taking place. Okay, generation of this antigen receptor for BCR, and more or less you have the same process for TCR except for a few changes, right? Okay, so one thing that you have to consider is that in germline DNA, there exist multiple gene segments which encode for single immunoglobulin heavy and light chain. Okay, segments. There are different segments which encode for these chains, these polypeptides that you have, which are eventually assembled to generate this tetrameric structure, where you have two chains of light and two of heavy, and then basically these antibodies are displayed on surface, and once cells are activated, so they are actually released or secreted out, and you can find them in extracellular spaces, right? Okay, so essentially during B cell maturation and stimulation, gene rearrangement, the gene segments are shuffled. Okay, so this is basically shuffling of these different segments that you have in variable regions, right? And because of this shuffling process, so you end up getting a coding sequence for the variable region of both heavy and light chains, right? So essentially, if you summarize this thing, so this actually means that chromosomal DNA in a mature B cell is not the same as you get this in basically in a germline. For example, in germline cells, for example, if you talk about sperm, ova, and basically during the formation of zygote, and eventually the different stages. So if you actually isolate cells from that stage, and if you compare the DNA of these cells with those that you recover from peripheral blood or secondary lymphoid organs, so the DNA is changed. And this change is happening because of the rearrangement process, right? Actually, this was shown by one of the elegant experiments, and obviously that was the first evidence that kind of suggested that probably this is how you generate a different kind of clones of B cells which can recognize one antigen or the other. Okay, so again, if you summarize these processes in a simplistic form, so you start from hematopoietic stem cells, you generate lymphoid cells. Okay, now these lymphoid cells, we are talking about B cells here. Okay, so they have to start this process of heavy chain gene rearrangement first. You generate, then basically once the heavy chain gene is rearranged, you start calling them as pro B cells. Okay, and then in these pro B cells, you start rearranging for this, where actually you have the partial rearrangement. So this is pro B cells. When you have a complete rearrangement, this is called a pre B cells. Now, pre B cells is the stage where your heavy chain is taken care of, and then now you actually have to start rearranging the light chain. Okay. However, at this stage, I also said I actually showed you with one of the different color coding that you can still at this stage get a surface expression of this BCR, which consists of rearranged heavy chain, but a surrogate light chain. Okay, so that you can start finding from pre-B cell stage. Okay. At some time point, this surrogate light chain has to be replaced by a canonical light chain. Okay, so that means this pre-BCR or expressing cells or pre-B cells, they start rearranging light chain, and then you mature, and then you actually become immature B cells. Immature B cells will have on their surface this membrane-bound IgM. Okay, so IgM basically is nothing but an antibody molecule, but it's tethered to the membrane. It has both heavy and light chains, right? After this, actually, you follow this process that we call as RNA splicing, okay, or alternative splicing. Okay, and this leads to the formation of another antibody isotype, again membrane-bound. Okay, you already had the IgM, membrane IgM, but through this process of RNA processing or RNA splicing, so you generate membrane-bound immunoglobulin D. And remember, so in, I think briefly we did talk about that the B cell receptor that you have on surface of your B cells, it consists of both IgM and IgD. Okay, they are going to have the same antigen specificity because the variable region remains same. The only change is for the constant heavy chain. Okay, and that basically generate a different isotype in IgM. So you have mu, and in in membrane IgD, so you have delta. Okay, now these cells have developed, and they actually are scanned through this process of positive and negative selection. If they were autoreactive, they were already eliminated, right? And now these cells actually can start populating peripheral lymphoid organs, secondary lymphoid organs. Okay, and they're waiting for this antigen to come across, and when they encounter this antigen, these activated cells become activated B cells. Become plasma cells, they start making antibodies, and these antibodies are secreted out. The first isotype has to be obviously IgM, a pentameric form of it. And then through class switching process, so you generate these different isotypes. Now, what decides which class is going to form? That will be decided by what cytokines are present in the germinal center. So this is basically the process where the activation of B cell actually is taking place in presence of one cytokine or the other cytokine. Okay, that will decide the isotype of antibody you are going to generate eventually, right? All these
Excuse me,
processes,
sir.
yeah.
Sir, could you please explain this slide once again briefly, please?
Yeah, sure. So you start from hematopoietic stem cells. These, if have to become lymphocytes. Okay, so basically they start down regulating some molecules, start expressing some molecules. What those molecules are, so we can talk about it in detail, or you can read some literature. Okay, but lymphoid stem cells will start rearranging first their immunoglobulin heavy chain locus. Once they have rearranged immunoglobulin heavy chain gene, okay, the product of this rearranged IgM, okay, the heavy chain of IgM assembles with what we call as surrogate light chain. Then it comes onto the surface. Then following this, so you replace this surrogate light chain with the conventional light chain, either lambda or kappa, right? And once you have a fully assembled immunoglobulin molecule, so this is displayed on surface because it has this transmembrane portion, so this remains associated with the membrane. And this is also required for signaling through antigen, following antigen recognition. However, at this stage, so you also have alternative splicing of this immunoglobulin, so that you start generating not only IgM, but you also make IgD. Okay, both IgM and IgD at this stage are present on the membrane, and this serve as the complex of the which actually comprise of the B cell receptor. So you also have the other molecules, Ig alpha and Ig beta and all those things which are required for transducing signaling in the cells. But at this stage, once you have formed membrane IgM and membrane IgD, so these are displayed on surface of these cells. Once they are displayed on surface, these cells are mature now. They have passed through the process of negative positive selection, so they don't react to your own antigen. They are only against foreign antigens. And now they can actually start coming to peripheral lymphoid organs, particularly the secondary lymphoid organs, lymph nodes, spleen, and so on and so forth, right? And they are waiting there for sensing the presence of any antigen. Okay, for example, either you are immunized or you encounter some kind of infection, or you start eating some food. Some of the antigens are also present in food, right? So they actually then start activating these mature B cells. Mature B cells become plasma cells. Plasma cells start making antibodies. And depending on what environment is present in the lymph node at germinal center site, so there is actually a particular site that we call a germinal center reaction. So that actually is dictated by what cytokines are present. Depending on, for example, if you have IL-4, so you start making IgG. If you have interferon gamma, rather if you have interferon gamma, so you actually make this IgG. If you have TGF-beta, you make IgA, and so on and so forth. So all these processes of class switching are dictated by the cytokine present in the germinal center in secondary lymphoid organs. Clear?
Yes, sir. Thank you.
Okay, so when people were trying to discuss or discover actually how this whole process of generation of antigen specificity is taking place, and how on the earth, so you can actually have a repertoire which offers you the ability to recognize and respond to each and every antigen present in this universe. See, well, you can say it theoretically, practically you cannot prove it, but apparently a healthy immune system has the ability to respond to everything and anything that has ever been formed or will ever be formed. Okay, so this is basically the estimated profile of antigen receptor diversity you can generate. Okay, and a lot of those clones have been sequenced to identify, to characterize that these are actually different unique clones, more than 10 to the power 7 or so have been shown experimentally. Obviously, with these new techniques of NGS sequencing and so on and so forth, so we now know this number is way higher than what was initially predicted. Okay. Now, question here they were trying to address was how you can explain generation of diversity in your BCR or TCR. Okay. So one of the very convincing thought process was actually given by or hypothesis actually was given by Dreyer and Bennett in 1965 at this time point. So no experimental proof was there. Okay. What they suggested was two separate gene encode for single immunoglobulin heavy and light chain. See, remember, you have a heavy chain, you have a light chain. If you talk about one gene, one polypeptide hypothesis, then you would like to think that there is one gene which encodes for heavy chain, one for the light chain. However, here what they were trying to suggest was that there are two separate genes which encode for heavy and light chain. That is for the first time they actually kind of suggested. After that, they said that even in the because you have to have this process of rearrangement where you can keep the constant portion as same, but you can keep shuffling this variable region in the antibody molecule. So apparently, so even within the same polypeptide, the constant region has to be encoded by something else and variable region by something else. Something means some genes. And then they kind of suggested that probably in the variable region, so you have rather several hundreds or thousands of these genes, which actually can be recombined. However, remember at this time point, so people actually started knowing a little bit more about molecular biology. And they kind of challenged this whole thing, that how on the earth this is possible. So yes, we know one gene, one polypeptide. We also know that from one gene, you can actually generate, you can have the alternative splicing, right? So you can generate a different polypeptide. But here you are trying to say that several genes encode for one polypeptide, right? So this actually was a very out of way proposal at that time. And remember, we are talking about 1965 at that time. Okay. And obviously, one for the constant regions. That's what they proposed. No data was there. Okay. After that, so even more complexity to this whole process was revealed, obviously experimentally as well. And then people actually starting to look at where these chromosome segment, where these genes are located, right? And they actually, eventually, this is a summary which kind of actually prove this hypothesis that the light chain gene, lambda and kappa, they are actually present on different chromosome. For example, in humans, so lambda chain is present on chromosome number 22 and kappa on chromosome number 2. And same is the case with mouse. It's a different chromosome, 16 and 6. And for heavy chain, so this is present on chromosome 12 in mouse, and human, it's 14 chromosome number 14. So apparently, so if they have to actually come together and form a product, so the rearrangement, reshuffling, all these processes have to take place, right? One more idea at that time was proposed, rather it was actually an experimental proof. There was this guy from Germany. So he actually proposed, rather he actually sequenced these different clones of B cells. And he showed that in variable region, you find a lot of uh, basically basically you have the polypeptide chain, right? So in only in N terminus of this, actually you have a lot of variation in amino acid usage, but constant region remained same. He presented this work in a conference, and he actually was the first one to show it. Okay, but probably either he was not convinced or he was not actually sure about his observations. So he actually passed through this slide very quickly, didn't even talk about this thing in detail, and because all these immunologists were gathered there, so they were actually trying to, the purpose was to actually find out a potential theory which might explain antibody diversity. Okay, and he has this data which show that in variable regions, so you have a different amino acid usage, right? But they said they actually found it very interesting that actually looks like we are getting somewhere to actually discover how this whole process of rearrangement and eventually how you can actually generate a lot of these antigen specificity on BCR or TCR, right? They said, could you go back to this previous slide? So once the talk was over, and he said no, and somehow he rushed through his whole presentation, and he left that meeting. But that actually, because all these scientists were sitting there, and they were all smart people, right? So they actually got an idea that probably this is the way to pursue. Okay, and eventually the whole field, when a lot of these different theories were given initially, so it focused analysis on looking at the molecular biology of these different chains that you have, the gene which are encoding for it, and that somehow eventually provided how antibody diversity could be generated. Okay, this is just one anecdote, so I wanted to share with you. Okay, so DNA rearrangement, it produces a variable region. And once you already have rearranged this variable region, so basically it then through alternative splicing of mRNA, so it attaches with the constant region and it gives rise to a polypeptide for either light chain or for the heavy chain, right? So essentially this is what it means. So you have one of the V's, one of the J's, it was shown for light chain, right? And then once you have rearranged V and J, then one of these concentration region for the light chain kappa in this case. So it was attached to this process through RNA splicing process, and you then formed this rearranged DNA, which can be transcribed and translated to form the light chain, right? Same is the process with heavy chain, except for that you have one more segment inserted somewhere here, which we call as diversity segment, D segment, VDJ, right? Okay, first proof that this rearrangement actually is happening was provided by Sumuto Onegawa. Experiment was, rather this was actually published in 1976. And at that time, actually, still, this is considered as a bombshell experiment. Because this was the first evidence, very convincing piece of data they collected to demonstrate that this rearrangement actually is happening. How did they do it? So they collected the DNA from germline, the cells which actually were in the germline stage, or they also collected the DNA from this rearranged liver cells. Okay, remember, so the whatever the genetic makeup you have, it's same everywhere, right? Obviously, you can get it from B cells to make it more convincing, and they did so, and the data actually is shown for the B cells here, right? So DNA is a huge DNA, just like that. So what you have to do is you have to actually cut it into small pieces. And in order to do it, you have to perform this process that we call as a restriction enzyme digestion, right? Depending on what sites are present, so you generate these different segments. Again, this was actually a very brave experiment at that time. So what they did was, so they performed the process of southern blotting. Okay, so this is called southern blotting, where you are probing DNA. And the probe actually is a sequence of nucleotide. And if it has complementarity, it will bind to it. And then you can actually say that, yeah, now you got the signal. Okay. All right. So they perform restriction digestion on DNA isolated from germline or where it was rearranged. That means mature cells, right? And generated a probe very strategically in, for example, J segment. Okay. For example, if you could have generated this probe in a different area, in constant region, for example, so you won't get anything like this, right? So generating of this probe was also very critical. So to actually convincingly, or to actually get some interpretable results. So they generated this probe against the sequence that you find in the J region. Okay. You perform restriction digestion and resolve this DNA from germline and from mature cells. Use this probe, and what they got was that if you have this DNA isolated from germline cells, so you get a band in southern blot, which actually was running very low. So that means, so here, so wherever this probe was binding, this was actually a short sequence, right? However, using the same probe, if they analyze the DNA from mature cells, so they found that now it actually has shifted. So that means now the actually fragment is larger. Although the probe is binding exactly to the same thing, but maybe this segment is attached with something else. And that is why, so you get a larger sized band. This actually convincingly showed that this rearrangement process actually is taking place. And then he generated a lot of these libraries to actually show that these are different clones and so on and so forth, right? Different sequences. But because of performing this experiment, obviously Sumoto Negawa was given Nobel, I think in probably 96, right? When was it?
I think so, 1984.
So whenever. So he was given a Nobel for performing this. So he wrote this paper to Nature. And because once you already have achieved a Nobel in one field, then you kind of think that probably there is no chance of getting another Nobel in the same field, right? Then actually he shifted his area. Now he's not doing immunology anymore. He's actually a neuroscientist and making big discoveries in neuroscience as well. Okay. Obviously, he's not going to get another Nobel, but he already has contributed big time. Anyways, so you understand what I actually explained in the previous slide? That rearrangement process actually was taking place, which they showed by isolating DNA from germline cells and from mature cells, and they showed that there is a shift in the size of this band, which was detected using the same probe, and this probe came from J chain, right? Rather, a J segment of in the it's the nucleotide sequence, right? So then now things were I guess kind of settling to say that yes, rearrangement is the process which kind of explains how you generate different antigen receptor diversity or the specificities, and depending on what is the number of these genes, so you can actually generate more or less diversity, right? So essentially, if you summarize it, so the organization is like you have the leader sequence right? Let's say if you talk about these light chains, kappa and lambda. So first is leader sequence, then you have the V segments, and then you have J and then constant. For heavy chain, leader sequence, V, D, J, and then constant. Okay, so this is for the variable region, and this is basically the constant region then is attached to it through RNA splicing process, occurring both for light chain and heavy chain. And this whole complex then is assembled to make a BCR, obviously also a TCR. Okay, so this is the organization of these different segments. And you can find differences that how these are arranged. For example, for lambda chaining, so what you have is leader V, J, C, right? And obviously then you have some pseudo genes, all these constant, right? And then again, for a different, here you have leader V lambda 2, here you have lambda V1, J, and all. The organization is different for lambda and kappa. Here you have leader V, leader V, leader V, right? And then a stretch of all these J's. Here J's are scattered. So this organization, as I said, can be different for different chains. But the eventual process is by shuffling of these segments and bringing one of the D's, one of the J's, and then once you have recombined this D and J, then one of the V's. And then once you generate it for heavy chain, you start the same process for light chain to generate V and J segments, and eventually this is combined with the constant region through splicing process, and you make the receptor. Okay, this is for the heavy chain. You have leader V, a lot of those V's, D's, J's, and then this different constant gene, the gene segment which encode for the constant region. So as you can see, the organization is like, first you have C mu and then delta. And these are the first isotypes that you form in differentiating, rather developing cells, right? And then you have this gamma, right? Gamma is the then antibody which you formed after isotype or class switching. And then you have epsilon, right? And then you have alpha. So because mu and delta actually are present in close proximity to variable region genes, so this is more efficient process to generate a receptor which carry constant region from mu or delta, mu and delta rather, so you have both of these. But the antigen specificity is decided by only the variable region, and it remains same for that C mu or delta, right? You have a question?
So what does the psi sign in two of the J segments signify?
It's a pseudo gene. Okay, so they discovered that the sequence is similar to J, but somehow this is not utilized in any of the clones. Pseudo gene.
Fine, sir.
Okay, variable region gene rearrangement. This is what we are talking about. So variable region gene, just go through this slide, just read it. It occurs in B cell during the maturation process in bone marrow. Heavy chain variable region genes rearrange first, then light. In the end, so in B cells, which contain a fully functional rearranged DNA sequence, right? Then this attaches with the constant region for mu and delta, and through class switching, so you generate a different isotype. You don't really call the formation of this IgD as class switching. Theoretically, you can call it, because actually it's a different class. Okay, but this has nothing to do with the activation profile of these cells. So this actually is part of the whole BCR that you get. Class switching, you usually you use this term when antigen stimulation is happening, right? Okay, more or less, we are actually talking about the same thing. Okay, so you have germline, heavy chain DNA, a lot of V's, D's, J's. Initially, you have D and J, and then following D and J, so one of the V's come and complete this process of rearrangement of heavy chain, right? You have already gotten this VDJ for the heavy chain, and through RNA splicing, basically either you make C mu or C delta. Okay, and then translated product will give you this nascent polypeptide chain, which carry heavy chain or delta chain. Okay, same process then has to happen for, well, the sequence we already have talked about. First heavy chain, then light chain, and then basically, obviously, the alternative processing and so on and so forth to generate the whole receptor. Light chain, again, one of these V's combined with J's and then with the constant, right? And then if this constant kappa is arranged, so you make this a rearranged segment, okay, which is actually then transcribed to make this transcript, and then you make the translated product as light chain, right? So all these processes that occur for every single transcript you make, as you know, for molecular biology, polyiodine deletion, and all those. So those are going to happen for the antibody molecules as well. Okay, but the key thing is this rearrangement process occurring at DNA level. Once this happens, so then you make this transcript, and after transcript, then rest of the things remain same. Okay, how basically this cutting and pasting is brought about. So this we are going to discuss in next few slides. Okay, so you have these specific sequences that we call as recombination signal sequences. Okay, these come in actually two forms. One is there are two kinds. Okay, responsible for this recombination process in BDJ. Okay, they are more or less constant sequence. The location of the sequence actually can be different, whether you are talking about V's, D's, or J's. Okay, so one type actually is where you have 12 base pair sequence. Okay, just look at this thing here. So these RSS sequence consist of the sequence are actually of nucleotides which are organized in a way where you have a sequence of this heptamer. Heptamer is what? Seven nucleotides, right? And then you have any sequence of 23 base pairs. Okay, and followed by this you have a nonamer sequence. Nonamer and heptamer remains constant. Okay. Once you have this, for example, this 23 base pair sequence interspersed between heptamer and nonamer, so you actually, because this is actually a bigger sequence, so actually it can undergo this two-turn process. Okay. If instead of 23 base pair, if you have only 12 base pair, so this sequence, whatever you have, this is a small sequence and it can only undergo one turn. So this is called one-turn RSS, and this is called two-turn RSS sequence. That is decided by the interspersed sequence that you have. In one case, it's 23 base pair. In another, you have 12 base pair. Okay, which actually are shown here with these triangles, right? So you have leader, V, and then basically one of these triangles you can see, right? And this is actually 3' of this V lambda, for example, right? For kappa as well, and for heavy chain as well, right? But for J, so they are actually present at the 5' end. And so this also actually gives you an idea that somehow this, because these two processes, so this have, for example, let's say 1-turn sequence. This is a 2-turn sequence. 2-turn sequence and 1-turn sequence will come together. So these actually are recognized by this recombinases, RAG1 and RAG2 enzymes. And obviously, so you have many more enzymes which actually were discovered subsequently, right? Okay, but the critical role actually is played by RAG1 and RAG2. Okay, so they actually recognize these sequences. They sit in there and they cut, and they bring up, as a result of this cutting, so they bring these segments closer. And this bringing them closer actually is going to generate the rearranged segments, right? So it's always a 1-turn and 2-turn rule. Okay, 1-turn cannot always combine with 1-turn, right? So you have to have 2 combined with 1. For example, in kappa, so this is a 1-turn here, so obviously it can only combine with a segment which has a 2-turn sequence. So this actually is a rule that kind of dictates that you have this specific recombination event taking place. So 12 can join only 23 and vice versa. Okay, 12 cannot combine with 12. And that actually makes sure that you don't have these nonsensical recombination events. Okay, so this is how the sequence actually is looking like. This is this RSS sequence. So you have, let's say, VLAMP. So this most of this analysis was done with light chain. Okay, and why? Because light chain is very small, and it really does not have this transmembrane nonsense, because dealing with a transcript which have a transmembrane encoding sequence, so it makes life difficult. So most of this initial analysis was done with light chain, right? So this is how these things are organized. So you have a sequence of nonomer, 12 base pair sequence, and then heptamer. And if it has to combine with the J, so at the N terminus of this J, you have to have this 2-turn sequence. And only then these recombinases will come, sit in there, and they bring these 2 segments together. Okay, so what we talked about was mechanism of variable region DNA rearrangement is catalyzed by these enzymes that we call as RAG1 and RAG2, right? And these are the one which are responsible for bringing these VDJ segments together. So this is what it means, and they actually recognize these sequence specifically, this 1-turn sequence and 2-turn sequences, right? And only then the whole process of this combination is going to take place. Obviously, when actually this cutting and pasting is happening, so you can always assume that this is not a very perfect process, right? Invariably, so this is a very highly energy driven process, and a lot of these rearrangements that are happening in your bone marrow for B cells and for T cells in thymus for rearranging this TCR and BCR, very non-productive process, but you have to make sure that you do not generate these TCRs which come out of these primary lymphoid organs, which could be potentially autoreactive. Okay, and this is the price you have to pay for that. So a lot of cells have to die in this whole process, right? So this imprecise joining can occur so that reading frame is not complete. And if the reading frame is not complete, obviously cell is not going to survive, it will die. And it was estimated that less than 1 out of 10, for example, rather 1 out of 9, they have to actually die, or rather reverse. So out of these 10 events, only 1 is likely to be successful. And obviously, once you rearrange this thing randomly, so you generate autoreactive clones, though these cells also have to die. Okay. So one more thing that actually I would like to do it now is, what time do we have? Yeah, so I think this is a quick video. So I want to actually run this video so that actually you understand this more clearly. This is hyperlink is not there. So I'll try to put it somewhere. So are you able to see this video?
Yes, sir, we can see, but
You can
audio-
see, I'll put it in the full screen, right? Yes?
sir,
Yes, sir.
yeah, it's not audible well.
It's not audible. So let's try.
That become joined together by a process called somatic recombination to make a functional gene. In heavy chain genes, there are three gene segments: the variable or V segment, a diversity or D gene segment, and the joining or J segment. Light chain genes, such as those shown here, have only two gene segments, the V and the J segments. Gene segments that can be recombined have specific sequence motifs adjacent to them, called recombination signal sequence, or RSS motifs. A protein complex containing the products of the recombination activator genes, RAD1 and RAD2, binds specifically to the RSS motifs. In this example, flanking a V gene segment and a J gene segment. The individual gene segments, to whose flanking RSS motifs the RAD protein complexes bind, are selected at random from a number of copies present at each gene locus. The RAD protein complexes bring together the gene segments to be recombined and cleave the DNA exactly at the junction of the gene segment and its adjoining RSS motif. The cleavage creates a hairpin of DNA at the end of the gene segments and double-stranded breaks at the ends of the RSS motifs. Additional proteins, DNA-dependent protein kinase, Ku, Artemis, and a dimer of DNA ligase and XRCC4 are incorporated into a large complex with the RAD proteins. These RSS ends are joined, forming what is called the signal joint, to create a closed circle of DNA that plays no further role in the recombination process.
Okay, so this video actually was old. Now we kind of know that these loop that you generate, they also have some role to play. Not well defined though, but people actually have suggested some role associated with them. Okay.
Hairpins at the ends of the gene segments are then cleaved. An additional enzyme, terminal deoxynucleotidyltransferase, or TDT, is recruited and adds additional nucleotides to the ends of the DNA strands. The other enzymes in the complex ligate together the two ends of the gene segments, completing the recombination process.
Okay, so it kind of make things easier to understand, right? So you can just copy paste this link in YouTube and you'll find it rather in this whatever the URL you have. You can go through this video again. But the process actually is recognition of this RSS sequence by REG1 and REG2 enzymes, one strand cleavage at junction of the coding and signal sequences, formation of these VJ hairpin, ligations, involvement of other proteins that they talked about. These are all having different functions. Creation of this hairpin, activity of these TDTs, which actually can add additional nucleotides. And then basically the whole process actually is, the whole sequence actually is ligated and repair actually is taking place. Following this, you have recombined whatever the V, whatever the D, whatever the J segment you have, right? And this process will keep on happening in these clones unless you get a productive recombination, right? If you have exhausted all the options of rearranging TCR or BCR successfully for heavy and light chain for BCR and for alpha and beta for TCR, and if you still fail, the cell has to die. Okay. So what is shown here actually is these kind of loops. They actually have been shown through this electron microscopy that actually in developing B cells, you can actually get these different loops. And this is more observed in cells undergoing the process of recombination, right? Okay, so I don't know how much more time we have. 2-3 more minutes. Yeah. So what is happening is because this is actually an imprecise joining. So many a time this is a non-productive, but this imprecise joining actually is also helpful to some extent. Okay. Because if you have incorporated additional nucleotides, or if you have deleted some, and then these repair enzymes try to repair and make it as a in-frame sequence of, for example, these 3 nucleotides, then you actually generate a new amino acid, right? And new amino acid means novel specificity. And this also actually amplifies the level of antibody repertoire that you are able to have, or TCR you are able to generate, right? So this process actually is helpful in creating more diversity, right? In this table here, it's shown that if you actually look at these different clones, they have different utilization of these. The sequence actually is different. Okay, this is a nucleotide, and if you actually translate it to amino acid sequence, that is also going to be different, right? And each one of those cell line actually has a different antigen specificity, right? So this actually is helping in generating more and more antigen specificity. It was also briefly mentioned about P and N nucleotide addition. Okay, and for this, obviously, when you generate this hairpin, so it has to be repaired with all these enzymes that was talked about in the previous video. And once you correct those hairpins, so and you perform this repair mechanism, so the complementary nucleotides are added on the other chain, and then you generate, for example, these new nucleotides, which can eventually encode for amino acid to provide you with novel antigen specificity, right? These are 2 types. These are called junctional diversity, okay? Because these are happening at the junction of D at J or V and D, right? The other approach actually is with N nucleotide addition, where actually the activity of this enzyme terminal deoxynucleotide transferase is very critical. So you can see that here, so you have only what, 1, 2, 3, 4 plus 4, and here 4 and 4 are there, but you already have inserted a codon which will encode for another amino acid to give you additional antigen specificity. And just by adding one more amino acid, you can actually change the antigen specificity of your antibody, right? Antibody diversity, P addition and N addition. Okay. The other process that we have to talk about is allelic exclusion, but let's not start this topic. We'll do it in next class. So questions, maybe.
So,
Brief questions, 1 or 2, maybe. Yeah.
so we saw in the video many additional proteins which are involved. So are all these proteins encoded by the same basal only, basal genome?
Well, yeah, obviously. So wherever these proteins with enzymatic function are required, they are induced to express in more abundance. But in other cells, you have the sequences, right? And for example, if you choose the right microenvironment, so they can be induced in other cells as well. But obviously, they are predominantly present in B cells.
Fine, sir.
Yeah. If no more questions, so we'll start this allelic exclusion and somatic hypermutation in tomorrow's lecture, and continue it, rather take it forward. Okay, so now I have to actually stop this.